it's a great pleasure to be here and fantastic to follow up from Bruce and the other speakers in the morning that already provided a very good introduction about stroke. But I come from a lab that study host response to stroke using a preclinical model. So we work with mice. Now, you should know mouse do not spontaneously develop stroke. No matter how much fat you give them, no matter how much glucose you give them, there's no clot in the brain. In fact, we have to surgically implant a thrombus into the brain, and we do that via two strategies. The first one, more recently adopted in my lab, is called a photothrombotic stroke, which Dan have provided a very good introduction, where we inject a photosensitive dye called Rose-Van-Gaal systemically, and then we shine a laser to a region of the brain at which we cause a damage to endothelium. Then there's a clot form, and we can see a localized infarct. In my lab, we specialize in a technique called intravital microscopy. What that means is that we can study blood vessel activity live in an animal. We do that in the brain via shaving the skull to a membrane-thin layer, so a laser can shine through, and we can watch processes in real time. Here, Josh will, in the audience, in the next session, will talk to you more about this. But what he can visualize is that there are platelet dynamics, very turbulent dynamics at the advancement of the clot. And surrounding this clot, we see these brain-resident macrophages, where we call microglia. They're highly active, of which they are interacting not only with themselves, but also with other cells within the brain. I'll leave Josh to talk to you more about this model this afternoon. The other model we use is called a mid-cerebral artery occlusion, where we surgically insert a monofilament through the external carotid artery, which rests at the start of the mid-cerebral artery. We can leave that filament for a certain period of time, and that's the beauty of this model, because we can mimic patients that had a thrombectomy or patients that did not have thrombolysis or thrombectomy. Here you can see the infarct development in either of which the mice have a transient ischemic event or of which they have a permanent ischemic event. But regardless of the model, if you take a closer look at the brain infarct and surrounding that brain infarct, we can see localized activation of microglia. Now, Brooke here noticed that microglia of an injured part of the brain are highly dendritic. They have a small cell body. Their dendrites are nice, long, and complex. On the other hand, if you find a microglia close to the infarct area, these cell bodies are huge. They have much less complex dendrites, and they are much shorter. And what we've also done is that we took those microglia out and not do transcriptomics, we did proteomics, because we really wanted to know what are the real functions of these microglia after stroke. And we found there's over 100 different proteins that were different between the microglia on the non-injured side compared to the injured side of the brain. Most of these proteins are related or associated with cytokine production, chemotaxis activities, and migration. What I want to talk to you about today is not just only host response to stroke in the brain. There's actually a lot of processes outside the brain that stroke cause. One of the clinical evidence for this is that a lot of stroke patients will develop infections after stroke, of which a big majority of them will die from this infection. A lot of these infections are highly prominent in the lung and urinary tract, and it really points to us that clinically, if you give antibiotics to these patients, no matter whether you're targeting patients with infection or targeting patients that have a huge infarct, it has not been effective in improving the outcome of these patients. What it really means is we don't really know what's happening to patients systemically after a stroke. Well, we've had some clues. We believe this comes about because the nervous system and the immune system are tightly regulated. They're bidirectionally regulated, in fact. And a lot of immune cells actually express adrenergic receptors. So receptors that can respond to neurotransmitter. Here you can see invariant natural killer T cells trafficking through liver sinusoids, so blood vessels of the liver. They are patrolling the sinusoids looking for danger for us, any bacteria. Now, these NKT cells are also highly abundant in a neurotransmitter receptor called beta-2 adrenergic receptors, and they can respond to the release of catecholamines after the activation of sympathetic nervous system after stroke. And what we can see after stroke, this video is actually playing, but this graphically demonstrate that very quickly after stroke, behaviorally, these NKT cells are not functional, and functionally, they're also not producing the antibacterial cytokine required to protect the host. So we believe this is one of the reasons why patients with stroke are susceptible to infections. Now, that was during my postdoc days, and when I was studying the liver, of course, when you open up the abdomen, you'll see the gut as well. I was trying to convince my boss at that time that, hey, I can tell a mouse that had a stroke by looking at the gut and those that didn't have a stroke. Why I can see this is because after a stroke, I see these bubbles happening throughout the small intestine, and every now and then I'll see one in the colon. Sure, Connie, go back to Australia and study it yourself. And that's what I did. So I took the gut out of mice after stroke, and what I found was there was a transient breakdown of gut barrier after stroke. This happens within 3 hours after stroke onset. Now, this gut barrier dysfunction is very prominent because it's similar levels to that of mice that have colitis. The difference is that colitis is chronic gut dysfunction. Now, if we take a closer look of this gut barrier, this is a very effective barrier that protects us from danger and what's happening in the gut lumen. It's made up of millions of epithelial cells that are tightly joined together with a specialized protein family called the tight junctions. Now, we can study these tight junctions by doing immunofluorescence staining of Z01, and obviously we can quantify this. And what we found shortly after stroke is that these tight junction proteins are either dysfunctional or lowly expressed, because that's what we see through immunofluorescence. And we predicted that this dysfunction of tight junction allow toxic material from the gut lumen to penetrate through the gut barrier and disseminate systemically and cause all these havoc systemically in patients and animals with stroke. Now, one thing when we publish this, I always have this thought, is it real? Is it true? Can you also prove it more? Because I always think, that's just a family of protein. Bugs are quite big. How are they going to squeeze through these holes? And the next question I got my student thinking about is, what if this dysfunction of tight junction not only is dysfunction of the tight junction, but the actual cell that holds the tight junctions together? So we performed the flow cytometry of gut tissues after stroke, and what we did was we defined cells that were at late stage apoptosis or apoptotic death or live cells using markers such as annexin 5 and 7-AAD. And within 24 hours after stroke, we see an elevated death of intestinal epithelial cell, not only in the jejunum, but most prominently in the ileum of the small intestine after stroke in our post-stroke mice. And then came along an honor student who was really brave and asked for a project from me. And what I got him to do is count every cell from the gut that was positive for cell death. Because not only, facts can have its, downfall, right? Because you don't know the location, the region of which there's intestinal cell death, but I want to show that it's real, because dissociation cells for flow cytometry can have its shortcomings. So what we did was we collected as many gut tissues as we can from our sham-operated and post-stroke mice, and we used two strategies to prove there was cell death. Firstly is tunnel staining, so this identified broken DNA. That signifies cell death, and the other one is cleaved caspase 3, so active cells cells that are actively undergoing apoptosis. We can identify there is increased apoptotic cells not only in the intestinal villi but also intestinal crypts in the guts after stroke. But why is it happening? Well, PhD student Catherine, who's now graduated, we know for a fact that a lot of gut barrier breakdown happens in gut diseases like colitis, and a lot of these happens because there's poor perfusion into the region of the gut. We next looked at whether this gut barrier breakdown after stroke is because delivery of oxygen or blood flow in the gut has been impacted by stroke. We I prompted to you, we do intravital microscopy in the lab. So we've set up 2 intravital microscopy of the gut in the lab. One is looking at the muscle layer of the gut, so the surface of the gut. The other is actually opening up the gut, and now we can visualize the villi. Here, the yellow beads or the green beads you see are microspheres. They are inert microsphere where you can inject IV into an animal and then use that as a surrogate marker for perfusion rate. So what Catherine had to do is track each of these dots and quantify how fast they were growing. In the cirrhosis side of the post-stroke mouse, we see no difference to the sham-operated animal in the speed of which the perfusion is happening. Surprise to us was that after stroke, there was actually increased perfusion into the villi of mice that had a stroke. We have no explanation from this other than postulating this is because of increased metabolism, because the mice are not eating anymore. Perhaps the gut is starving for energy and wants to grab as much energy from the bloodstream as possible. Maybe that's why there is actually greater perfusion in the gut after stroke. A very recent paper that just got published in Nature Cardiovascular Research examined gut after stroke, but not just epithelium or perfusion. They started to look at a region of the gut that is high in, sorry, not a region of the gut, but a structure of the gut called the Peyer's patches. These are basically lymphoid organs of the gut that governs gut immunity. What they found after stroke is that these Peyer's patches shrink. And this is, again, within 24 hours, within hours after stroke. And when they did light sheet microscopy, they found that these Peyer's patches were very high in GP1B platelets. And they also have identified there are clots happening in these gut microvasculature in the Peyer's patches. And they postulate this is why there's poor perfusion. The cells are then also dying surrounding within the Peyer's patch. The senior researcher from that lab, we met in a conference just like this, and I told him what we can do, and he told me what questions he has to answer. Light sheet microscopy is amazing because it's clear, it's transparent tissue, but it doesn't allow you to see action. What we can do in intravital microscopy is that we can watch perfusion happening live. Now here we've set up the Peyer's patch intravital microscopy. This little pimple here is the Peyer's patch, and we can watch perfusion happening within this microvasculature. So this is really just to show off we can do this. We haven't got any data regarding this yet, unfortunately. We'll put in probably CD49B or GP1B to check whether there's platelets after stroke. One of the key questions we have also in the lab are all stroke the same? Do all stroke patients develop infection? We know they don't, but what makes a stroke patient susceptible to infection? And we performed a retrospective study in partnership with our clinical stroke clinicians at Monash Health, where we looked at 500 patients over a 12-month period, and we found out that it doesn't really matter whether the patient had an ischemic stroke or hemorrhagic stroke. The risk of infection is the same. However, as you probably predicted, the older the patient, the more likelihood they will develop infection. And we can recapitulate that in our preclinical model, where we set up a model such that we see equal survival rate between our young and aged animals, because we don't want to just study survival bias. But of those that survived or lived in our aged animal, there was a 100-fold increase in the spontaneous the risk of spontaneous infection rate. And we predicted this is because perhaps these animals had an impairment of antibacterial function in the lung. Now, what is the antibacterial function in the lung? One cell that we are particularly interested in is called neutrophil. We've heard a couple of talks about these. They are really quick and fast responders for infection and injury. They're highly migratory, they're fast. They're armed with antibacterial weapons. Now, we can study how well they move after stroke using an intravital microscopy technique, whereby we can pull the cremaster muscle of a male mouse, of course. And how we set this up is that after stroke between young and aged mice, we expose the cremaster muscle to a gel, which is high concentration of chemoattractant CXCL1, and we wait and see how far these neutrophil extravasate from the vessel towards the gel, how fast they go, how far they go, and whether it was a directed migration. And what's better to show you this than videos? So we got contestant blue and contestant green. They're off on their marks, and you can see blue is winning the race, and they know exactly where they're going, whereas the green dot, sorry to say, but really just circling around a site that is distal to where they're supposed to go. So I can show you graphs, but I think this is more visible. Sure, but that's the cremaster muscle. What about the lung, where all the infections is happening? So we've set up the lung intravital microscopy in the lab, and I'm probably safe to say we are the only one in Australia that is doing this. We can now visualize the live microvasculature of the lung, and what you see in green are the neutrophils. First thing you see is that the vessels are not straight. They're all over the place. It's a network of microvasculature. So studying migration is really difficult here. But when you have a really, really patient postdoc, they can calculate or count how many cells they were crawling, how many were stationary. In young mice that respond to stroke, we found that a lot of them start stopping. But in aged mice, it seems like this neutrophil is ignorant to the distal injury in the brain. One other thing I told you about these neutrophils is that they're highly phagocytic as well. So they can move to the bacteria and gobble it up. So we try to study this again in vivo at the functional pulmonary microvasculature. We injected mice with a dye called Frodo. So these are pH-sensitive bacterial microparticles. Once these bioparticles get into a cell, it moves to the phagolysosome where the pH drops significantly, and then the dye will light up red. And again, I'll show you this by video. Hopefully you can appreciate in the young mice, within probably 7 minutes, you can see the red dots coming up. On the other hand, at the same time, you see a lack of this happening in our aged post-stroke animals. So really just counting the red dots that's popping up in these neutrophils here. I'm cautious of time, but this is one of my last slides. Inherently, we do see a deficiency of neutrophil phagocytosis for aging, but this capacity is further reduced if the animal had a stroke. Now, I want to put everything together. Of course, I can't tell you everything that we are doing in the lab. I really wanted to show you is that we can use intravital microscopy to study a lot of these processes, all the way from the brain, liver, lung, the gut, and different parts of the gut as well. And probably the most important slide for this talk is thanking all the people that have contributed, my clinical stroke partners, my folks that are super knowledgeable about gut and epithelium, inflammation experts, core facility, and everyone that I've highlighted from my lab. This can't be done without funding, and I thank them graciously.